Okay, welcome, and we're going to talk about today where the immunocytes come from, where the immune cells of the immune system originate from. Um, the cells all originate from a very common central place, and that are the bone marrows. There's the bone marrow, and the take-home message from this section is going to be three basic points. One, all cells of the immune system originate from the bone marrow, particular bone marrow cell stem cells um, are critical. These are so critical that if you lose these cells, you can't generate either immunocytes or immune cells or blood cells. Two, that these stem cell differentiation is guided by a variety of growth factors they depend on that they come into contact with. Three, the effect of growth factors on a cell is determined by the type of growth factor receptors on the surface of the cell. So that effect it may be obvious, but those growth factors are receptors are the key communication, the way that the cell receives the communication from the growth factors. Now, the cells of the immune system, there are leukocytes. These are the white blood cells that can be divided into accessory cells and lymphocytes. Taken as a whole, accessory cells comprise the largest percentage of leukocytes. We can see these here. Neutrophils. Neutrophils are about 50 to 70% of those cells. Remember, they're key, really important cells in their antibacterial effect, ability to combat bacterial infections. Next are monocytes, which are basically blood-borne macrophages. Eosinophils, which are in the tissues, which screen for potential tissue parasites, as well as basophils. And these accessory cells activate lymphocytes, they carry out and increase phagocytosis, and they secrete powerful effector molecules. So these are actually the largest part of these leukocytes. Now, the process by which red and blood cells originate come from, the process that they're generated is referred to as hematopoiesis. And it has several characteristics. Red and white blood cells originate from these specialized cells in the bone marrow called pluripotent stem cells, by this process referred to as hematopoiesis. These are responsible for the production of erythrocytes, granulocytes, monocytes, mast cells, lymphocytes, and megakaryocytes. Basically all the cells in the immune system and all the cells of the blood cells. Now, the pluripotent stem cells are very important, are the most important cells. There's not many of them. There's about 1 in 10,000 bone marrow cells are these cells, these stem cells. So they're by far one of the lowest population of cells in the bone marrow. But what they're characterized by, their ability to do three things. One of those is differentiate along multiple pathways, so they can, in the proper environment, differentiate into other cell types. Two, they can self-renewal, so that when they divide, if they divide, they can make another stem cell just like it, so they can renew themselves. So you don't run out of the stem cells. And three, they can proliferate when needed. So under certain conditions of stress, when the immune system is really being activated, these will proliferate into additional cells, additional stem cells, into additional differentiated cells. But there is only 1 in 10,000 bone marrow cells are stem cells. Now, when you get a bone marrow transplant, the most important part when a bone marrow transplant, for example, if a patient is undergoing chemotherapy or radiation therapy, which destroys their bone marrow, so that they can have the bone marrow from another donor put in, transplanted in. It's the cells, it's these stem cells that are the most important, and there are not many of them, but they can be infused and will home to the bone marrow and begin differentiating, self-renewing and differentiating into the into the immune cells and red blood cells. So key point here is that once a stem cell has undergone differentiation, it is referred to as a committed progenitor. So it can't go back. So progenitors can't de-differentiate back into stem cells. These cells have no ability for self-renewal. They go along their progenitors, so they'll go along a path to their final endpoint, to the cells that that are required. For example, there'll be a progenitors, lymphocyte progenitors, lymphoid progenitors may go along a path to become B cells, for example, or macrophages. It just depends on that environment. The path of differentiation taken by a progenitor is determined by the following microenvironment factors. These are critical factors that are in the environment of those cells. One is the stromal cell support. So these are stromal cells. Stromal cells are really, you might consider them kind of nurse cells. They emit cytokines. They provide nutrients. They provide an environment for which the cells to differentiate into their to the cells that they're ultimately supposed to. The microenvironments are important for the quality and quantity of the growth factors. And that's important. The number of these and the amount of those growth factors are important to determine what cells they differentiate into. And 3, the differentiation and proliferation is controlled by growth factors called cytokines. Those that act on lymphocytes are known as lymphokines. So it's sort of a specialized, lymphokines is a specialized type of cytokine that acts on lymphocytes. So overall, these growth factors, you'll hear this term referred to as cytokines. Now, growth factors or cytokines are peptide factors, which affect cells, and lymphokines are a subset of these peptide factors, which affect lymphocyte action. There's more than 20 are known to be produced by T cells. There's a variety of these different, oh man, there must be maybe over 30 different types of interleukins, different types of these factors. They're produced by different cells. And they are small proteins, small peptides. They're generally glycoproteins, and they can be detectable in the serum. So, for example, if someone lymphocytes are being activated, you can go into the plasma and you could see this activation by monitoring the amount of these cytokines that are that show up in the blood. Now, when cytokines, while the cytokines may differ in structure, the receptors fall in distinct families. So you can think about these cytokines in the receptors. One way to think about them is the types of receptors that they interact for. The cytokines themselves can be different in these. In other words, there can be different structures of these cytokines that will then interact with receptors that have similar kinds of structures. And they can fall into basically 4 classes. One is the hematopoietin family, and these cytokines that fall into this I've got a group of sampling of those, anywhere from IL-2 to erythropoietin. All of these fall in this. These are the numbered IL-3, 4, 5, 6, 7. There's the immunoglobulin family, which IL-6 and, for example, IL-1 fall into. There's a mistake there that should not be. You can strike that out. IL-6 there. For there, it should be 5 and 7. There's the tumor necrosis factor, which is TNF. It's a TNF family, which is again the tumor necrosis factor, CD40 and FAS. And then there is the chemokine families, which are IL-8, MIPS, and all the chemokines. There's many of these types. And some of these actually have become drugs, particularly like erythropoietin. We'll see these, and this is really where the biotech industry got its start, was from actually being able to produce discover and produce these kinds of cytokines and use them as drugs. Now, one of the first families is the hematopoietin family, and these are characterized by very interesting by basically being homodimers. And so if we look here, I'm going to stop this right here. We look here, the green and the red are the two arms of the receptor. The blue is the actual cytokine. This is the EPO, the cytokine, erythropoietin. And what happens is these, let me just get my pen here. Let's see. Okay. So what happens here is you have one side, you have one of these is one part of the one monomer, and another the other one. And these come together to form the binding pocket in which the cytokine is going to sit in. And then the membrane is along here, and then these signals are then transferred. What we're seeing here is crystal structures of the cytoplasmic domains, but then these go through they will go through the membrane and emit their signals through their membrane transmembrane domains into the cytoplasm. And so the key thing here is that these two parts come together. The cytokine EPO, when it binds the dimer, and so if it binds a monomer, it causes the dimerization. And so dimerization of the receptor, bringing together the green and the red arms of the receptor, are hallmark of this kind of the hematopoietin receptor family. They basically come together and they form a binding site for the cytokine. Now you'll notice that, let me see if we can start again. Kind of go back here a second. Yeah, okay. So you'll notice that, for example, here, as we rotate this, the cytokine binds deeply into the pocket, and then it binds this arm, the red arm, which is identical to the green arm. I just have them different colors with this part of the molecule, whereas another part of the molecule binds to this part of the receptor arm. My point here is that, as you can see here, the cytokine itself is asymmetric. In other words, it's not the same on this side as it is on this side, and so it actually interacts with the two arms differently. So it binds to the red part of the EPO receptor, the one arm up here with this part that's sticking out, this little loop, where it binds to the green, the identical green arm of the receptor down here. And so there's symmetry between, in other words, there's symmetry between the along the receptor, along this axis, the actual binding of the cytokine to the binding site is asymmetric. And so you can see it doesn't bind. It's quite a bit. It binds differently. Just to repeat, the green and the red are identical, and they come together to form the binding site and for the cytokine. And the cytokine itself binds asymmetrically to the two homodimers. So the receptors that are in this, those cytokines that are listed that are in this family, their receptors are similar in the sense that the receptors have this characteristic of being homodimers. and it's when the cytokine binds to it that the signal is transmitted by conformational changes into the cell. Now, the other one is the immunoglobulin superfamily, and in this one, IL-1 is an example, and it has it's called the immunoglobulin superfamily because you can see it's the different subunits here of it. If we take a look here, have this beta sheets. There are these beta sheet domains that are very similar when you think about it to an antibody. In other words, which has these beta sheet domains, these constant region domains. It's made of largely these beta sheets. In addition, it has this curved part where it forms basically the binding site for the cytokine is this curved part where it binds, the cytokine binds in as it docks into this binding site is established by this curving of the receptor, such that it binds, such the cytokine can fit into this cavity, which you can see here a little bit clearer right there. You can see the cavity that it's bound into. That's made up, and then these domains, these three domains, make up that binding site when it interacts with the cytokine. So this has a one domain. This type of receptor family has a one domain makeup in which then the signal is sent through the cell in this direction down into the cytoplasm when it binds. Now, the other family is the TNF family, or the tumor necrosis factor family. And this is unique in that the receptor is almost like a rigid pole. It sticks in through the membrane, and it does have beta sheet and loop structures that are stapled together with disulfides. But it's interesting in that it binds the surface. It binds like one side of the cytokine. So actually, the other part of the cytokine is does not interact with the receptor, as we can see here, but just one face of the cytokine binds to the actual receptor. And you can see here, for example, here's these loops here that are binding into this receptor, into the receptor. Now, this has enabled researchers to think of designing basically peptides that can mimic this, or small molecules that might mimic this interaction, that might be able to block the effect of TNF, tumor necrosis factor, because it actually interacts with discrete molecular interactions in several places along the receptor. And so that has been ongoing avenue of research. If you think about it, if we go back one to this one, we go back to this family. It may be very difficult and to design something that would block this interaction or cause the same interaction if it's a small molecule, because you need to be able to interact with this entire active site surface, or binding surface, which is very large, and so from a design standpoint, that becomes quite challenging. But for tumor necrosis factor, there are small molecules. There's been quite a bit of work to try to design ones that can mimic or be antagonists of this interaction. The last family are the chemokines, and these are the chemokines are unique in that they are transmembrane, 7-membrane helix transmembrane. Let's see if I can get my pen here. Sorry. They're transmembrane. What that means is that the membrane, as you probably know from GPCRs, the membrane spans this region in which the chemokine is going to interact at some spot on the helix bundle, and the helix bundle, and transmit the signal through the membrane into the cytoplasm. So this is the outside of the cell, this is the inside of the cell. And these are helix, these are in this case 7 helices that span this, and this is a very, very common framework for GPCRs, for example, and other types of receptors in that class. The conformational changes upon binding are transmitted through this membrane to factors on the other side, like G proteins on the other side in the cytoplasm. So all those that are in this class have these receptors have this general type of structure. Now these are, again, alpha helices, and they're particularly good at forming structures, being able to be stable in membranes and packed together with themselves. They tend to be, as we've said before, alpha helices tend to be the most unstable, least stable secondary structures, but they're quite stable in membranes. And that's one of the reasons that you see these in membranes as opposed to beta sheets. And they also are very good at transmitting conformational signals through the receptor, allosteric signals through the cell, through the membrane to the cytoplasm. So when we're looking at, that's just a kind of a brief look at the different types of receptors, the cytokines and their structure, how we relate the cytokines as receptors, not based on their function, but just based on the structure of their receptors. They can be grouped into these 4 different kind of classes. Now they all have different functions. These receptors and cytokines have different functions, and that function for hematopoiesis must go on in the blood, must go on in the bone marrow. So if we start with a stem cell can self-renew, and it can make another cell. It can make another one. We can divide into another stem cell, and in the process, a stem cell can also, in the presence of cytokines like IL-3 and granulocyte macrophage colony stimulating factor, can also differentiate into myeloid cells. And myeloid cells can further differentiate in the presence of these different cytokines. IL-3 and GM-CSF and EPO will lead to erythrocyte progenitors, and more EPO in that environment will lead to erythrocytes. If you have TPO, then TPO leads to megakaryocytes and then on to platelets. TPO is one of the lowest concentration factors of cytokines that we have out there. But in that presence, that's how you get from platelets. So the point here is that you need, for platelets, for these myeloid cells for these progenitors to eventually give you platelets, you need EPO and TPO, thymopoietin, and then EPO is needed for erythrocytes. So you can see here if we used EPO as a drug, that would, if we were to inject someone with EPO, that could lead to more erythrocyte development. Now, in the presence of different combinations, for example, IL-3, GM-CSF, and IL-6, you'll form granulocyte progenitors, which leads to neutrophils or monocytes, given whether it's either granulocyte GCSF or monocyte CSF, and then on to macrophages. And so this is the arm that will then, these progenitors, the stem cells could, in the presence of these excuse me, cytokines, there's no EPO here, there's IL-3 and IL-6, give you granulocyte progenitors. If it's IL-3 and just GM-CSF, you'll go the route of producing basophils, eosinophils, and progenitors, and on in either with IL-5 eosinophils or IL-4 basophils. This gives you just a flavor of the different requirements that the cytokines have the cells have, that differentiation pathway has for the different cytokines in that macroenvironment. Now, what's key in that microenvironment in the bone marrow is what the quality and the amount of these cytokines. So the amount of these cytokines that they find themselves in will lead then at any of those spots to different cells that are produced. Now, if we don't have GM-CSF, then we're going to go, the stem cell can differentiate into the lymphoid cells. And this is a little bit different. And then there's no GM-CSF, there's no EPO, no TPO. There's only the IL-7 or IL-4, so the interleukins, and these will either differentiate along a path to B cell progenitors or T cell progenitors. And IL-7 is important for the B cell progenitors on to B cells, and IL-4, as we've seen, can go on to plasmacytes. Now these, of course, this differentiation here for B cells in the plasmacyte into plasmacytes, that occurs. That's going to occur in actually in other organs. So the progenitors produce the B cells, and the B cells will leave the bone marrow and go to the secondary organs, and we'll talk about that later. That'll become clearer. For T cell progenitors, we'll go to thymocytes. These are going to leave the bone marrow, and they'll go to the thymus to where we'll see deformed T cells. So, so this is everything up to here happens in the bone marrow. So this is in the bone marrow, and everything out here then happens. These happen outside the bone marrow. The plasmacyte development and T cells. T cells in the thymus and plasmacytes generating the lymph in the spleen and the lymph nodes. And we'll see that later. Now, the cytokines can form, have been used as drugs, and they are typically their properties. They're like I said, glycoproteins, or proteins which have glycosylation on them. Interleukin-1 is an endogenous pyrogen produced by a variety of cells, immune cells. It activates Th cells and reduces to release lymphokines. It's sort of a broadly activating immune stimulant, and it stimulates a lot of different effects among the immune cells. Fever, acute phase proteins, it just does a lot. It's a major inflammatory protein. And so there is also interest in how to correct this, potentially for inflammation that's out of control. Interleukin-2, as we've seen before, IL-2, is important for T cell activation, is necessary for T cell activation. And it's also about 15 kilodalton glycosylated protein. This is acidic. Excuse me. Acidic. Acidic as well. This is slightly acidic. Interleukin-3 is produced by basically colony. It's basically a multi-colony stimulating factor. It's produced by lymphocyte progenitors. It's a much larger protein. It's 20 to 26 kilodaltons, and it's glycosylated again. And this stimulates production of numerous cells, as we saw in this chart. It's involved in the myeloid cell production, just as a major factor there in getting the myeloid cells. Interleukin-4 is produced by activated T cells, and that stimulates proliferation of B cells and T cells. Interleukin-5 is also produced by T cells, and that stimulates IgM production, IgA secretion by B cells. It stimulates differentiation in eosinophils. I'm just giving you a flavor for some of these, that they're produced by different cells and they can have different effects. Some large, some very like IL-1 and IL-3 can have large effects on lots of different cells, whereas IL-2, for example, and IL-4 has specific effects on specific cells. IL-6 is produced by monocytes, transferred fibroblasts, uh, um, lymphocytes. It's produced by a lot. It's produced by a number of these types of cells. It's large. It's one of the most important cells in that or cytokines in that it leads to really induced activation of the immune system, and it stimulates production of, as we'll see, HLA. This is actually MHC1, as we'll see later. This is another name for expression. Stimulates IL-2 production, and just has a wide range of effects on the immune cells in proliferation and differentiation, particularly, for example, macrophages. And you'll see this. This is one of the major cytokines for inducing potentially systemic cytokine activation. Tumor necrosis factor we saw before is produced by macrophages. It kills tumor cells, synergizes its effects with others. This is a really interesting molecule because the reason it was discovered basically because it was noted early. It was noticed that people who had bacterial infections, actually their tumors could shrink, and so it was later found years later that this factor that's produced by the macrophages during the immune activation response was largely responsible for that, and that's led to a lot of interest in it as an anti-cancer agent. GCSF, or granulocyte colony stimulating factor, we've seen is produced by monocytes, endothelial cells, and again another 15 to 20, 18 to 22 kilodalton glycoprotein, has different glycosylation patterns, stimulates the production of neutrophils from these as we've seen from committed precursor cells, and activates mature neutrophils to produce ADC and phagocytic activity. So some of these can also point here, can stimulate production of the new cells, differentiation, as well as their activities, the cell's activities, activity for some of these cells as well. Granulocyte macrophage colony stimulating factor, or GM-CSF, which is a drug, activated. It's produced by activated T cells, macrophages, endothelial cells, a lot of different cells, and immune cells as well as endothelial cells. Again, it's a glycosylated protein. It stimulates production of neutrophils, eosinophils, and macrophages, as we've seen. So this is a really important one if you're low in these. If a patient is low in the amount of, they've undergone, for example, chemotherapy, and their amount of neutrophils are quite low, then you can give GM-CSF, for example, to increase the amount of neutrophils in their plasma, in their blood, so that they can be protected from bacterial infections. The macrophage colony stimulating factor is a little bit more selective for growth of a few cells like eosinophils, monocytes, and macrophages, and it activates it increases the ability for macrophages to want to go under phagocytosis, and also ADCC. Now, one of the molecules I talked about before has been one of the most important molecules in biotechnology is erythropoietin, or EPO, and it's procreate epogen has been names that it's been sold under. It's produced by the kidneys. It's a 125 amino acid glycoprotein, and this is very selective. It stimulates the production of erythrocytes, and under the right conditions with thrombopoietin, it can stimulate platelets, the production of platelets. So this is used. This can be used. If you lose, for example, if you have kidney failure or you lose your kidneys, then one of the problems that you have is that you can't produce EPO, and so therefore your erythrocyte levels can diminish, or you can lose the amount of erythrocytes that you're producing, and your blood the amount of blood erythrocytes can go down. And so as a consequence, patients that have this, who are undergoing, for example, dialysis and all, have kidney failure, are given EPO to increase their hematocrit. And that's a long-standing drug now. It's got to be 25 years that that's been used. IL-7 stimulates the proliferation of precursors of B cells and T cells, as we've seen. And IL-8 is a neutrophil attractant protein, and it induces lymphocytes to be attracted into areas where it's produced, and there'd be areas of infection. And as I said before, thrombopoietin is similar to EPO in structure and is the most dilute biological active substance that we've known of. It rapidly stimulates the differentiation, proliferation of megakaryocytes and platelets. In fact, one of the key works was to identify it was done down the road at the Mayo Clinic in a collaboration between the Mayo Clinic and Genentech. You're going to hear more about these. This is just a taste of these, what they do. You're going to hear more about these in more detail as biotech products and biotechnology in the biotech when you take your class on biotechnology. But this just gives you a flavor of the ability of these molecules to induce different cell differentiation patterns upon needed in their production, and to also potentially be used as drugs themselves to carry out those same effects if patients are low, for example, erythrocytes or neutrophils. So here are 3 questions. These are 3 review questions. So you can go ahead and look at those. And you can stop the video now, take some time and answer those questions. So I would stop that now. And then the answers will be on the next page. So that's it for today, and that's the end of the lecture. And what I'd like you to do is to have viewed the lecture, viewed this material. It'd be good to have this lecture material viewed so that you for class on so that you'll be familiar with this this material for class on Wednesday. Okay.